EREC faculty presenting their research regarding their program. And today we are glad to have Dr. Herman Sandoya, who is a lettuce breeder here at the EREC Hort Sciences Department. And he's going to talk to us about too many traits, too little time to breed lettuce. With that, Dr. Sandoya, floor is yours. Thank you.
Good morning, everyone here in the Everglades Research and Education Center. Also, I see people across the state connected to this seminar. Welcome to our house, the Everglades Research and Education Center. And these seminars have been prepared by the seminar committee to present what we do here at the Everglades Research and Education Center and to foster possible collaborations. So this seminar has the title that Too Many Traits, Too Little Time, because as a plant breeders, we had to take care of every single trait possible because, you don't want, for example, a lettuce or a cherry that is resistant to a disease, but it doesn't taste well, et cetera, right? Over a year ago, oh, that goes, okay. Yeah, there it is. Over a year ago, when I was asked to present my seminar to the Horticultural Science Department for tenure and promotion, I thought, what's the best way to present a program like this one, right? And I used shapes and sizes. As you see, diseases is probably what I've been working the most. And that's the reason why that one is bigger. But I've been doing work also in post-harvest traits, heat tolerance, nutrient use efficiency, other issues such as nutrition in the crop, the greenhouse production, et cetera, that are not as important so far, but require some of the work that we do in plant breeding, right? And before I'm going to present a few of those, the work we done on this. But before I do that, I just want to show you all the people who has been involved in this research. Lots of students, postdocs, and the different topics, as you can see here. Some of them move on into bigger and better things and are working now. For example, Catherine is working in Cornell University, and Gustavo is working in North Dakota, and Liz is doing her PhD, and some of the postdocs are working for the industry, et cetera. And why diseases? Because the crop is actually attacked by many pathogens, as you can see here in this picture. Some of these pathogens are problems to producers here in the Everglades agricultural area, like downy mildew. Specifically, my program looks a lot to bacterial leaf spot, fusarium wilt. So in the past, used to be a lot more corky root than now, I believe, and that has been a little bit eliminated because the flooding practices that happen here in the Everglades agricultural area. We also see cercospora leaf spot, especially at the beginning of the season, at the very last of the season. Greenhouse people see it all year long, and it's a very complicated disease to eradicate. We don't have Brazilian wilt, for example, but it's another wilting disease similar to fusarium. Some point we did have a lot of sclerotinia and diseases like, other diseases are not listed here. When I joined this, the group of faculty at the EREC, I was asked to work mostly in bacterial leaf spot because that's what growers don't have any way to control, right? The pathogen at that point was called Xanthomonas campestris, the pathovirus vitians. What the pathovirus means is exclusively to lettuce. That's what I understand. So, but back in 2020, the pathogen was reclassified as Xanthomonas hortoro-pathovirus vitians. As you see the two names, that means that's the same pathogen that's causing this disease. As you can see here in the picture, this is a romaine lettuce infected by bacterial leaf spot. So what some of the growers have been doing when they detect that really early is destroy the crop areas around, so that way the disease doesn't spread, right? It's not like I recommend the practice, but I think it's working well so far. There are some copper-based fungicides that people, the literature says helps field with bacterial leaf spot, but it's a very complicated disease. Therefore, resistant cultivars are the best option. So what this pathogen is a very diverse pathogen. So what we know, for example, race 1 was identified in Florida. As you can see, those were, they are in that blue color, L44 and L7. I think they were isolated by Dr. Pernesny here in the room today. But the newer ones were actually collected by our postdoc at that time in 2019. So we sent them to the pathologists to understand what races they were. So I think it's a little complicated to say they're race 1, but at this point they were considered race 1. But there's also race 2 and race 3. One was detected in Canada, the other one in California. None of those have been detected here, but it's something that we keep an eye on it, just in case that ever happens. So we've done some work related to that. The good news is there is resistance, right? The California group started to do some of the work for race 1. As you can see, the color relates to the race where the isolation was collected. In the case of PI 35801-1, that was identified as resistant by my predecessor, Dr. Liu. And then later on, we did a better screening of, with the California group for all the races, for all the isolates considered race 1. The ones here in Florida, we did that part, and they did the race 1 in California. So we found this PI here, 667690, to be resistant to it. And then because I've been doing lattice work for a while before here, one of the work that was done in California before my time here was to identify a race resistant to race 2 and race 3. So the paper finally was put together, almost effective, I hope, and where we report that these two group of accessions are resistant to races 2 and 3. The issue with those ones are Lactuca cereola, which is a wild type of lettuce, and it will take us longer to do the breeding. However, we have made some grosses in the project that my student Byron Manzanero is conducted under my supervision. We did a lot of work on the genetics of Lactuca because this has not only Lactuca sativa, which is a cultivated lettuce, it also has a lot of Lactuca cereolas and virosas and many other wild types. And however many work we do, there's like 1, 2, 3, 4 biparental populations. That means that we cross one lettuce with the other. And this GWAS population is a genome-wide association studies mapping population with about 500 accessions that were also screened for different isolates of the pathogen. We find a common area, which is in linkage group 2 or in the chromosome 2, the lettuce genome, which is great. We think it's a single dominant locus that controls the resistance. And for that, for us, for breeders, that means happiness because we can transfer the resistance pretty easily. And now we have resistance to race 1 of the pathogen. Race 2 and race 3 seems to be more complicated to understand, and the work needs to still be conducted as we move forward in this group. As I mentioned to you before, we did find PI 667690 to be resistant to race 1 in Florida as well. What we didn't know is the locus that is present in PI 35801-1 that a previous postdoc in this group called XCVR, Xanthomonas campestris vitians resistance, was similar to the Xanthomonas resistant one in another cultivar, the cultivar La Brillante. We did find later on that was the case, but we don't know anything about these ones, these newer sources of resistance, because we haven't done mapping yet. Maybe I shouldn't say that. Byron is doing that. But what I quickly did was to assess whether this is going to be a different source of resistance because we want to integrate as many locus possible, especially if they are different. So we do a test that is called an allelism test. We cross resistant by resistant. Normally, we will cross something that is resistant by susceptible, but in this case, we cross resistant by resistant. With the assumption that if we find one susceptible individual for every 16 plants that are resistant, we will say maybe they are no link. So we didn't find that. So that it says to us that it probably is the same type of resistance. However, this is how the population looks like. We have developed so many different families There is a plethora of different characters on on those families. In addition to that, PI 6690, we also found it to be resistant to Fusarium wilt, which is actually a good thing for the program because this disease showed up in 2017. It was a wilting disease. We didn't know what it was until we did the test. We collected several isolates from the first ranches where we saw the disease. And then Jesse Murray came into the story with his master, and later on Gulnoza, Isamudinova, did a lot of the screenings as well. And first, what Jesse found was the pathogen seems to be race 1. That's what it was mostly reporting in the United States. Then we did significant numbers of screenings. I don't even remember how many, maybe 10, 12. But we screened everything possible, including wild types. As you can see here, this is a wild type of lettuce showing the disease. Then we did crosses of the resistant lines that we have, the resistant lines developed by the USDA to our elite lines in the breeding program. So in addition to do most of this work in greenhouse, because we didn't have fields, only one area where we could do tests, we took some of the most promising accessions to California, to Arizona, and Yuma, and Welton, to see if they were going to be really resistant in the field conditions. So far, we found 2 that are really common to be resistant across these 3 different places. 6182, which is the lime in the breeding program here, that it wasn't developed for Fusarium. And again, PI 667690. All the things that we've been looking at, especially this one, our student called it pinwrig or pinking, was because growers brought us this. This is no lettuce, this is escarole with this disorder. And also we did some work in shell life for lettuce because, as you know, our lettuce traveled, I guess, all the East Coast all the way to Canada in some occasions, right? The first thing that we needed to do was to evaluate somehow quickly the shell life of lettuce, and then Catherine and Steve Sargent in the horse science department did work in a test called accelerated shell life testing, right? Instead of storing the lettuce 0 degrees Celsius or 4 degrees Celsius, whatever the packing house uses, they did a 15 degrees Celsius to accelerate the process. But we also screen lettuce for less pinwrig and longer shell life. So there's Catherine there doing some work here at the Everglades Agricultural Area at that time. I'm going to show you for the test only pictures. So she put lettuces at 15 degrees Celsius and at 5 degrees Celsius. As you see, at 5 degrees Celsius, the deterioration is less compared to the 15 degrees. And then depending on how many days the lettuce gets a value of 5, that is multiplied by the number, so based on the transpiration rate, and gives you the number of days that you believe that lettuce is going to be having good shell life. So the 2 examples here is PI 35801-1, which is the resistant to bacterial leaf spot, bad shell life, by the way, and Okeechobee, which is or was a commercial cultivar highly planted here in the area. So there's a difference in the shell life, as you can see in the picture. These results here are the different germplasms we tested. We picked a group of romaine, greasehead, or iceberg lettuce, butterhead lettuce, leaf lettuce. We even include lightning lettuce in the test. We did experiments for 2 years, or 2 seasons, basically. I shouldn't say 2 years, 2 seasons. Here in the EAA, some of those were stopped because of the famous COVID-19 pandemic. But in the end, we were able to get the data needed. On top, you see the ones for less pinwrig. So basically, we were looking at those ones that have the less value here. This is how Catherine evaluated, scale of 0, no pinwrig. She induced the pinwrig, by the way, to 5, where there was a lot of pinking in there. For shell life was a little bit different because what post-harvest specialists, they considered that 9 was the best. So your lettuce is good, you will buy it. compared to one that you don't see pictures of like this here, but something like this will be a one. There's no edible anymore, right? And then you transform this into values to have days of shelf life for your lettuce. Both have been published. This one was the last one published a few weeks ago, in fact. So, but besides that, one of the things we did in collaboration with Young-In Kim in the Horse Science Department was to understand if there's a way to reduce pin rate. So Young-In in her lab, she uses these metabolites to test many different things. And what she did with Catherine was to test cysteine, melatonin, natrigenin, and serotonin to see if you could reduce pin rate. Melatonin has the potential, in fact, has shown some good results, as you can see here in these pictures that you don't see pin rate with melatonin either, romaine or iceberg lettuce. And there's actually a patent application on that. In addition to that, for shelf life, Dr. T. Liu and Dr. Kevin Bexey and the Horse Science and Environmental Horse Department work together. And especially T believes that the senescent genes are associated to shelf life. And this was the work of Katie Chase in Kevin's group. What she did was to basically take the samples, do an RNA sequencing experiment, and understand what is they will find those genes associated to shelf life. This one's one of the orthologs characterized in Arabidopsis, and you can see here, this gene here promotes leaf senescence. That's the reason why it's more expressed on 6184, which was a lettuce that had poor shelf life. Similarly, this one delays, and then you're gonna see that Okeechobee has more expression of that gene. And this last one promotes leaf senescence, and again, 6184 is higher expression, or had a higher expression of the gene. Well, enough about post-harvest. We're gonna move into heat tolerance because it's really hot, as you can see here in Florida. That was the work of my master's student, Hannah Miller, who was doing the heat tolerance work. And the reason why we, besides talking to producers about the issues they have, they also told us, I wasn't here at that time, when the season used to be starting in September all the way to May. And these days, some growers start in September when possible, but reality is more October to April. So there's a reduction in the season, right? And what heat does to lettuce is actually a cool season crop. Is first, there's a reduction in germination, then it's bolting and flowering. I hope you can see that this picture here where you already see a little bit buds of lettuce flowering, or these long cores, that it means that the plant is bolted because of the heat. There are other consequences because of the heat as well, like tick-borne. It's not necessarily heat related, but its presence is accelerated by the warmer temperatures. So one of the things I did at some point was to put together a graph of the soil temperature based on the Florida automated weather stations. And I went all the way from 2011 to 2021. I haven't done 2022 to 2023 or 2023 to 2024 season. From September all the way to April, as you can see, these two are actually these first seasons here where I put together. Haven't gone back more than that. But you see, soil temperature is pretty high in September for lettuce, probably. 86 is one of the tests that Hannah conducted to understand seed germination. Bambino, which is a very sensitive to heat germplasm, doesn't really germinate compared to one of the lines, 6179, that it does germinate at that temperature. Then Maria Medina came into the story. Maria, now works at the ERC, and did a lot of testing in growth chambers, actually. The test hasn't been done in soils yet. That's something we want to do next. One of the things we did, and she did a lot of that, was to test 373 lactuca germplasm from our germplasm bank. We chose wild types, plant introductions that are currently at the USDA, cultivars that are known here in Florida, or other cultivars that are no longer used by the industry, and then the UFIFAS breeding lines. We went all the way hot, super hot. 21 just was the control, but we put this all the way to 36 degrees Celsius and 97 Fahrenheit. So the temperatures are in both for you to understand. And we did see a reduction when you get to the 97 point. A lot of the Florida germplasm seems to hold a little better compared to others. But based on that selection, we selected 29 germplasms. I'm gonna show you only results from 8. 4 of the worst and 4 of the best on germination, but we did went back because reality, we don't have soil temperatures at 97. And as you can see here in this graph, we found at least 3, maybe 4, that do well all the way to 32 degrees Celsius or 90 degrees Fahrenheit. So, and that might be a solution that we need to look forward, in addition to include tolerance with the whole plant, include tolerance to the heat and in the seed. But one of the things we did, though, was to screen lettuces in warmer environments for the most part. You're gonna see in the next a figure of what I'm talking about. And we started with unreplicated screenings to test the most we could. We did about 120 different lettuces. And then based on those results, we narrowed down the number and went to 48. 36 from those screening and 12 of the controls were tolerant and sensitive identified in California. Once again, from the work we did over there. That lettuce was planted in the San Joaquin Valley and near in the south of California as well, just to, have that high temperature on those ones. So the controls are here, listed here, in each of the different types, as you can see. Some of the pictures from the last experiments we did with Roth Farm, for example. This is Cultivar and Boy, Red Dog, and Little Gem. So, and this is where we started. So these are the ones that are, this is your current season, or what I believe is the current season. And this is where we start testing the unreplicated tests. So we did about 6 of those. Of course, we had some tests in the middle of the season to compare, when lettuce gets good conditions versus what it doesn't get good conditions. And then these are 8 more or 9 more experiments that we've done in replicated tests all over, trying to cover every single spot possible. And the other thing that you see here is we done in the sand and also in the muck because this is where the lettuce is produced. But in the sands, because as you can see here, this is actually the new growing zone maps that was released a few months ago. Florida, as you go from all the way from the south to the north, we're in different zones for growing plants. The northern you go, the little cooler it is. So we gave it that different the temperature range to see what exactly we're gonna get. We don't know yet. We haven't gotten the data, analyzed the data. But just sneak peek of what's happening just in bolting, because bolting is one of those most informative characteristics for heat tolerance. You can see, just to show you the controls, the I-34 2479 was sensitive in the Californian environment. It's also very sensitive here, so we get a lot of bolting. King Henry didn't, which is great. King Henry, in fact, was developed from Tolgus, Maine. That was developed here at this center by Dr. Guzman, by the way. And what we want to do with this is to explain the tolerance, right? What we plan breeders call the genotype by environment interaction, because we want, when we develop a cultivar, to be useful for as many seasons possible and as many locations possible. So we take in data not only in the tolerance characteristics, but many other ones, just a few that you can see here in this slide: head weight, core length, plant height, head width. There are many other ones that are not listed. Environmental characteristics, day and night temperature, soil temperature at different depth, et cetera, et cetera. And then one thing that is for us to do, we haven't done it yet, we are just ready to go, is to do some sort of sequencing in those 48 lettuces. We're gonna be able to do that with a new set of markers. We have that were developed by breeding inside at Cornell University. And we're going to be able to locate where the genes are in that case. So stay put. This is a collaboration with Diego Jarquin and the agronomy department. Once we analyze the data, we hope to update you on this. But one thing we did realize when we were doing a heat tolerance experiment was that night temperatures influence heat tolerance, right? This was, a test was actually just done to see how lettuce was doing in the sand. And one of the things that the extension agent at that time was concerned was temperatures in Florida are too warm, right? Let's do the same cultivar successions, grow them in different areas. One in Sun City, Florida. The other one was in Bearsville, Georgia, where you can see the temperatures. The biggest difference was night temperatures, right? 61 versus 73. So these were, seeded at the same time, transplanted at the same time. The difference was that month that was growing in the different locations. So then Hannah came and did, from the selections at that point, what we were doing, we were testing these lettuces, pick 4 of those. Manatee, that is well known by growers here, 6183, one of the lines in the program. Bambino, very sensitive to heat, and 6179. This is what she did. This is what the picture she put. She did these experiments in grow chambers. These experiments were not in the field, not in the greenhouse. It was a completely controlled condition. Then she used 25 to 15, 25 Celsius. Sorry, I didn't put the in Fahrenheit for you to understand that. But that's what we consider as the control, right? And then started stressing lettuce, and we went to 25 completely, and then 35 to 20, 35 to 25. And then she did something that she wanted to do, and we supported the idea to be, during the day cooler and at night warmer. So there was a lot of changes in morphology and marketability, of course, but she also took photosynthesis data and many other characteristics, including biochemicals, because there is a theory that is changes in sugar in the plant. So we have data, we're working in those manuscripts as well. In addition, there is alteration and energy metabolism, a paper that was just published on specific genes that are related to heat tolerance. So at this point, I would like to move to what's currently happening, right? These are the people, some of the people you know here, they're here in the room. Diseases, Byron Manzanero, post-harvest Jesse Murray, and then Brianna Haynes, who's joining as we speak. So she starts next week. So as Kelvin Alorgi, and we are trying to hire a master's student to work on heat tolerance. And Jairo and Shatna that are here, they're working on the nutrient use efficiency part. So this is the work of Jesse Murray. One thing that we didn't show you, I didn't show you in the past was we measure some nutrients in lettuce. What to see first is people will say, I want to pay more money for your lettuce because your lettuce is more nutritious. Some growers promote that, especially in the West, right? We measure tocopherols, phytoquinones, and ascorbic acid, basically vitamin C, vitamin E, vitamin K. Especially phytoquinones are very abundant in vegetables. Any greens that you eat, collards, broccoli, whatever green that you eat, you're having those ones. Does lettuce has enough? No. But it turns out that lettuce is one of those crops that people consume the most in the United States. It's only actually after potatoes and tomatoes, then lettuce. So there was differences, and one of the concepts in the science part was, in collaboration with Gilles Vasek at the Horticultural Science Department, is that Arabidopsis plants, the mottle plant, that has more of these compounds and more resilience to problems like heat, post-harvest, et cetera, et cetera. So that's one of the things that Jesse is doing now, testing some of those lettuces to see if there is actually a relationship in a cultivated crop like lettuce. He is trying to understand which of these metabolites are also related to that tolerance so we can use also for selection when we have to. Eventually, what he wants to do is to understand the genetics of shell lice based on the nutrition, on these antioxidants. Byron is all bacterial lead spot in these seasons, and one of the things that he's done already is to understand what genes are differentially expressed in resistant lettuces. We had two types of resistances, as you remember, PI 35801-1 and PI 667690, and some others that are not that resistant, but still good for the industry. Also, understanding the structural biochemical analysis of these plants, because we believe it's not the same mechanisms. And he has started a program for including resistances to races 2 and 3. So we have several crosses of race 1 by race 2, race 1 by race 3, and race 2 by race 3 in preparations if that ever happens. Because as I said before, these are like tucaceriolas, and it will take us longer to do the breeding. So better to start early. In addition to that, what he's doing is to understanding what is crops and weeds in the area can host the pathogen. So stay put for that research coming up. Then the phosphorus efficiency, you can see, I just want to acknowledge Gustavo Cruz, who started this work. Jairo Arcos, also in the room, continues to do that. And he wants to, again, understand that genotype by environment interaction that happens when lettuce is under different rates of phosphorus. In addition, try to assess roots, because that's where the nutrients are absorbed by the plant. And you can see here, specifically in this picture, this is what was not considered efficient to phosphorus, and this is where it's inefficient lettuce to phosphorus. There is not much differences on the roots, on the ones that are efficient, compared to the ones that are not efficient. So there's more work that coming up on that. It's a new methodology to screen lettuces, and he's gonna be doing that. And also understanding the genetics, in addition to what genes are differentially expressed on lettuce for phosphorus efficiency. So those are the students. As in the past, people call the students to come here and explain, so I decided to explain that on their behalf. But at the end of this presentation, if you have any questions, feel free to ask them. They will be all excited about tell you what's going on in there. So, but because we had too many traits, as you can see, and too little time to do breed lettuce, one of the things what we want to move forward is to integrate these diseases instead of selecting for one, disease for more than one at the same time. Or to understand if this nutritional compound can provide you some protection for post-harvest or for, stresses like heat tolerance, et cetera, et cetera. I believe there should be a common resistance. This is the lettuce genome. This is a figure I didn't put it. This was my supervisor in UC Davis, Richard Mitchellmore, who gave me this figure. This is the lettuce genome, the 9 chromosomes of the lettuce genome. And where you see there is a mark on red is where major resistant clusters are together. Each one of these ones are different type of genes, and we have shown that some of those are common to provide resistance to different pathogens, for example. So this is the case. Once again, you saw PI 667690 resistant to VLS and Fusarium wilt on lettuce. So we had a mapping population that different plants that you saw, the different colors, shapes, red and green. And 158 of these families were tested by Byron. And they were all resistant to VLS because, two resistance combined together, good. But it happens that 53 of those ones were also resistant to Fusarium wilt lettuce. The rest were susceptible. In addition to that, Liz did a screening for Cercospora because Dr. Ray told us that we need to work on that since he started to see it to be more complicated. And it seems like at least these two are resistant as well. You're gonna see that in the next slide. In addition to that, we have a pathogen has many races, the case of bacterial yeast spot. This is the publication that is hopefully being sent back to the journal by this weekend, where you can see that some of those accessions are resistant to different races, right? So all that thing complicates the work of the breeder. So, but the new techniques like genomic selection, we can use these models to integrate multiple diseases and multiple races to understand if we can select those plants that are going to be resistant to those issues that affect the crop. One of the things that I wonder is, could heat-tolerant germ plants be susceptible to diseases? As I mentioned to you, I seen it at the beginning of the season in the EAA. This was a picture taken in a farm nearby by Liz, where the plant was very susceptible to cercospal leaf spot. As you go into the season currently, you don't see the disease, but as we had heat-tolerant germplasm, my worry is that some diseases might be present, right? Good news for the growers, they have good control methods. So, but greenhouse producers, for example, they do not, because there's no labels allowed in greenhouse. Recently, we visited a farm with Anna Mezzaros here in the room, where all the crop was gone by cercospal leaf spot, basically. So this is the data, preliminary data that Liz generated in my lab during her time at the master. You can see PI35 and PI66 has some resistance. So some of the lines that actually we developed, not for cercospal leaf spot, but for BLS, they also seem to be resistant, but we haven't done the test yet. So, but what matters the most in the program at the very end is the development of the germplasm. So that's my mission as a plant breeder here at the University of Florida. I use these different iceberg colors. They are not real. Probably the only real is this one, just to show you how advanced we are in the generations for breeding, right? So when you're the F8, F9, F10, that's kind of like a finished cultivar ready for release, right? So just to show you where we are for bacterial leaf spot, we're all the way here. In fact, these were the lines I inherited from my predecessor because PI 3581-1, and we took different approaches, including backcross method or a pedigree selection method again. And now the lines look like this. I'm not saying they are great, so we had to test that, but they look more like romaine as they were intended to be. For Fusarium, we are less advanced, of course, because the disease was first seen in 2017. And for programs like heat tolerance and the ones we are actually crossing BLS lines with Fusarium lines, together, we're in the very early stage. So this is Amanda Carroll, who I want to acknowledge for making all the crosses in the program very successfully. Last year, we did a preliminary test with few of the lines that are resistant to BLS. We went to Chuck Over, that you all know, in sandy soils, because he goes beyond when the season ends in here, to see how tolerant those lines were to the heat, basically. We know they are resistant. They don't look the best, but they do have, short course. It's the industry is interested on that. And we're gonna do extra text as the new seasons come in to narrow down and to pick the ones that we really like for the release. The one here was one of those crosses we produced with PI that is not resistant. Not every single thing is gonna be resistant, but it was liked by greenhouse producers. So we maintain it, we keep selecting it because, well, it might not be useful for field production, it might be useful for greenhouse. This is Chuck's comment. You can read them. He called them variety. BG means Belgrade, produced in Belgrade in 2022, 1106. The license is small. It doesn't get to the market, but it was slow to bolt. It means it may have some heat tolerance as well. But the other thing that worries me a lot as a plant breeder is how are we going to select these many traits, right? We had to develop phenotyping methods to measure especially those challenging traits, right? For heat tolerance, currently we base on the yield only and few other characteristics like bolting, maybe tick-borne, but there might be other ways to select this, right? The picture down here is actually the reflectance that is measured with this machine here, and it shows you the differences in different plants, basically, right? The different colors have differences in different plants. Normally, we start the season in October. That's when we go plant because we're gonna risk a lot of our seed. We don't have a lot of seed, in fact. breeder seed is very little at the beginning. And then we transfer those plants into the greenhouse all the way to March, maybe April. And then that's how we do it to have better seed. Because if we do this, we start, for example, December, the flowering and the seed production goes into the warmer period, and we don't have good quality seed. Sometimes you see lack of germination because of that. So for that, the Plant Breeding Endowment and the Dean Research provided funding to buy this new toy here, which is a field spectroradiometer. We're just about to setting it up and then start measuring differences in plants. For example, under heat, no heat, less nitrogen, more nitrogen, or less phosphorus, more phosphorus. In collaboration, this work is going to be collaboration with Kevin Wan from the GCRC and the AV department, who is a specialist in phenomics. We know that remote sensing can measure differences, right? So the example here again is wheat cultivars that researchers measure with this machine to understand which one is. So they can select, for example, if they're using it for crosses, which background I want to select. Or the one here is different crops at different rates of phosphorus, and the researchers find the differences, as you can see here in these pictures. So while there's already too many traits and too little time, we also have too many new threats to lettuce production. And we need to continue breeding lettuce and other crops for longer sustainability. So the work that you see here is just in the starts. For water use efficiency, Sandra Guzman in the Indian River Research and Education Center is measuring some of those with our lettuces. We want to start drought and salinity tolerance because it might be an issue here. And as you saw in the fence presentation, some of the insect resistant because it's related to viruses. I do want to acknowledge all the collaborators, some of those, as you can see your name here, sorry. At the University of Florida and beyond the University of Florida, I do a lot of work with the people in the West because lettuce is a highly cultivated crop in that area. A lot of the funding that came to my program, maybe some of those is not there yet, through the different programs you see, especially crop block program, especially crop multi-state, and a lot of also through the University of Florida as well. I do want to thank the support of UFIFAS, especially the Everglades Research and Education Center, the Horticultural Science Department, the Florida Vegetable Exchange, Station Agents that I get to work with, Anna, Bob, Christian, Francisco, Gary, Jonah, Hannah, Lorna, Liz, Vanessa, and Wanda Laughlin, who helps me with experiments in greenhouse at North. A big shout out to the Plant Breeding Graduate Initiative that have provided funding to train now a third PhD student, Gustavo Cruz. Jesse Murray is a PBGI student, and the new one, Kelvin Alorgi. And of course, thanks to the lettuce breeding team for all your support. Thank you. Any questions? Yes.
With the variety profile, most of those are Western varieties. Do you intend to try to incorporate some of those lines into your program? Because I think it would be very useful, and maybe you've done this already, to have some of the same information related to the principal varieties planted in the EAA, or in particularly in South Florida, sand or muck, as it's relevant to the growers. I know you had manatee in several of the trials, but most of the principal varieties were not included, but maybe you've done that work already.
Yeah, so the question is related to, is we plan to integrate all the germplasm that has been identified on the West into this program? Yeah, the answer is yes. So that's the reason why we tested them, we tested them over there, my time there, but now we tested it here because it's different heat, different environmental conditions. even though those experiments were done in the lower part of the states, I believe they still had a little bit more light than we do. So, but they do well for the most part. There were very few exceptions that something that was considered tolerant wasn't that tolerant. Yeah, so, I always ask you, what's the type of letters you want? One question, one comment at one time from another grower was, we want to have this type of iceberg, right? So I had that into account on what could help you in your industry. Any other questions? Avery.
Yeah, it was really interesting about the post-harvest work that you're doing, looking at shelf life. Have you done any work, or is there any way to increase shelf life of the pre-chopped packaged lettuce that you see in the store a lot? It's more of a convenience, and I know the shelf life is typically less, but is there a way to?
Yeah. So I particularly haven't done it, but it was done in the West. Most of the test was in that was called modified atmosphere packaging, because it was the lettuce was chopped, then was sort of the whatever they were using to put in the bag, store in the cold, and then see how they lasted for that specific environment. At the beginning, when I just got here, it was the first year I got here. We sent few lines of the program to all the way to Canada. I had the data, in fact. The data has been used as a preliminary data, but it's not being used for anything else because we haven't done any other work on. But we did send the lettuce all the way to Canada, and they chopped the lettuce, and it's been done. Yeah.
Herman, really interesting seminar. Thank you so much for all the work you have done. I was curious, you talked about melatonin to reduce. How would you apply that?
So the way this is done is, first what they needed to do was to understand how that was going to be dissolved, right? And they use like water, DMSO, and many different things, I don't remember from the top of my head what other components they use to dissolve the melatonin, and then just apply it to a specific concentration. But one of the things, so far what was working better was DMSO, and they need to find other ways to, in fact, apply it without becoming a concern, right? Melatonin is great because it helps you sleep. If you have problems sleeping like me, I'd be happy with it. In top of that, one of the things we want to do, and I need to talk to maybe Paul and everybody in the grower community, is we apply those ones, can that one increase shelf life as well? So that's one of our next steps in this project. That's why Brianna Haynes is coming to play, to see if we can, apply those compounds, see if we increase shelf life as well. Not just from the breeding perspective, but also from the management part. Oh, questions on Zoom? No questions so far yet. Yes.
Thank you, Herman. It was a really nice overview of what you've been doing and your lab is doing. Just a question about, because I'm still not familiar with the whole process. So what if University of Florida, per se, you find a promising variety or a cultivar, a heat-tolerant cultivar? How much time does it take to get to the market, or would it be available to the grower?
Mhm.
How soon?
Yeah, so in the bacterial yeast part, what we need to do now is narrow down the numbers first. We have, I don't know, 400 lines, because I don't discard anything, in the advanced stage. But after that, you also have hundreds and hundreds and hundreds, right? And once you narrow it down, what you need to do is regional trials to see where your line performs well. Then increase it, clean it for off types, and then release it. So that's our job for the next 3 years. And I'm hoping that we can release a cultivar by then, because that's what the Foundation for Seed Producers want me to do. We could release them as a lines now, but they want me to put our cultivar there.
And let's say if it's really good for heat tolerance, but maybe it's not as good for certain diseases or something like that, would you still proceed with that and push it because this is really good for heat
Yeah,
tolerance?
yes. And in fact, there are some lines that were inherited in the program that I never found records on what they were bred for. And, I didn't even show them there. Those ones are even more ready than the bacterial spot program. I continue selecting them to adapt them to the conditions. Most of those were romaine, not many icebergs, because there were not many iceberg lines left in the program. Now we have made more. And but it's something that is, for example, heat tolerant, it doesn't have the resistance. In the case of BLS, specifically, that's easy. That's what I said. We love when I show you that genetic map, only one area to be responsible for resistance because we cross, we select it, and good to go, right? What you improve further is that adaptation, that shape, that what the grower needs, et cetera, et cetera. So what we want to do, and we'll continue to do, is just increasing other things, crossing to more things. We have a group of elite lines that we call. So we incorporate any other trait coming into the program quickly somehow, a few years down the road. Yes.
Thank you so much for the presentation. I enjoyed learning about your program. And I'm curious to know what's your perspective on doing common or conventional breeding, like screening, crossing and screening, versus when you do transcriptomics and then you have hundreds and hundreds of high confidence candidate genes. Can you tell us a little bit about pros and cons and speed of the breeding process?
Yeah. So, at the end of the day, a lot of the transcriptomic work that you're talking about is done because we're university professors. We need to generate that science, right? I don't know of any program yet that uses the transcriptomic data to select, unless, it's a single, you suspect it's a single inherited characteristic, and you finally rely on the gene you identify to say and develop markers for that. What genomic selection doing is having many markers instead of one for those traits that are very complicated to select, right? And it's always a challenge for us to do multi-trait loci breeding. And that's because, in a transcriptome profile, you identify a gene. If you are lucky to identify one, you identify many. Like, how are you going to select those ones all together, right? Probably genomic selection I think, is the answer to that. Because, you evaluate too many plants, and if you evaluate too many plants, if you had a model that tells you which one to pick, in what conditions to pick, perhaps that's the way. Yeah. Any other questions?
There's one question on Zoom. Do you need to introgress genes from wild species? If yes, do you need to rescue embryo?
That's a good question. We do need, I think we've seen it. So there are some resistance in wild types. And I don't think we need to rescue embryos for the ones that we identify. Because Lactuca cereola crosses well with Lactuca sativa. It's easy to recover offspring. What is not easy is to identify the plant that has the characteristics of the cultivated lettuce versus the wild type. But if you were to think about the resistant, for example, I'm sorry that I use resistant a lot because you saw my program is mostly diseases. Virosa or even Floridana, there is a Lactuca floridana. In fact, I'm very interested for heat and salinity. Maybe because, we don't know how well they're gonna cross. Oh, one more question. Okay.
No, I want to say I'm really glad you're still using some of Dr. Guzman's materials. It's just a testament of how breeding takes a long time, and Dr. Guzman worked so hard. And for those of us who are lucky who knew him, it's a testament of what he's done. So thank you. That's all I want to say.
Yeah. So sadly, I didn't get to meet him because, I'm the The anecdotic story on that was I got the job in 2016. I moved here in October, and I think he was working until August. 2 more months and didn't get to meet him. All right, well, thanks everybody for attending the seminar. Appreciate it.